Welcome to Department of Medicine Grand Rounds. I'm Brad Hill, a professor in the Division of Environmental Medicine, and I'm happy to introduce Dr. Sanjay Srivastava, who's also a professor in the Division of Environmental Medicine and distinguished university scholar and a fellow of the American Heart Association. Dr. Srivastava got his undergraduate and graduate training from the University of Lucknow in India, with a Ph.D. in chemistry. Is that correct, Sanjay? And then went to University of Texas Medical Branch for his postdoctoral work, which he completed in 1998. Through this time, he worked a lot on oxidative stress and products derived from oxidative stress, and he continued that work into his faculty appointment. And I believe I first met Sanjay in 2002, and he had just received his first R1, and really he hasn't looked back, and this is almost $60 million of funding later. He's been highly productive. He's published around 150 papers in high-impact journals and trained numerous graduate students and postdocs and junior faculty. And he's really heading up the Superfund research program here at UofL and has recently been awarded another R01. I think he probably has another one in there somewhere, right? That's going on. So I don't want to take up too much time, and I'm happy to introduce Dr. Sanjay Srivastava.
Thanks, Brad. Good morning, everyone. So today I'm going to talk about a project that I've been involved with for the last 2 decades, and that is looking at the effect of environmental exposures on heart disease, diabetes, and inflammatory disorders. This is, Jason, this is not working.
Oh, there we go. Here we go.
Okay. So as we know, diabetes and heart disease are prevalent worldwide. More than 200 million people just in the US, India, and China have diabetes. And worldwide, heart disease is the leading cause of death, primarily through myocardial ischemia and stroke. And if you look in the United States, the environmental exposures, even at in a smaller community level exposure, you can see that there is an association of environmental exposures with the cardiovascular disease and metabolic disorders. And essentially, about 150,000 people die every year prematurely because of environmental exposures from the heart disease. If you look globally, then again, is prevalent throughout the world. And environmental pollution accounts for 9 million deaths worldwide. And out of that, 6 million deaths are attributed to just the air pollution. And of course, cardiovascular disease accounts for most of them, as 4.8 million deaths are associated with the inhaled exposures and cardiovascular disease. So what are the sources of these environment pollutants, and how do they affect the cardiometabolic disease is a big question. And we get these chemicals from various sources. It's because of industrialization, urbanization, deforestation, heat stress, agricultural sources, and all of these things lead to the exposures to chemicals of different varieties, including the pesticides, metals, volatile organic compounds, and so forth. And primarily these molecules, one way or the other, affect the endothelium, and that cause either the endothelium activation or endothelial toxicity, which leads to vascular inflammation and later on to thrombosis, which leads to MI and stroke. So if we look at the exposure to environmental chemicals and the mechanisms of the disease, it's a very complex phenomena. The toxicants could have a direct effect on the target systems, like I talked about vascular endothelium, for example. But at the same time, several of these toxicants could affect the lung, could affect the brain, or if they are ingested material, could be affecting the liver. And then we can have autocrine and paracrine effects, which could lead to systemic inflammation, endothelial activation, thrombosis, and the sequel of events associated with myocardial ischemia and heart attack and stroke. And compounding these environmental exposure problem is the emerging threat, which is actually not a threat anymore, a reality, which is climate change. And climate change affects human population in many ways. And of course, cardiovascular system is also at the risk of the environmental exposures. We have known from the recent literature coming out that climate change directly contributes to human emergencies in the form of heat waves, or actually fluctuation of the weather with spikes in heat and cold waves, wildfire, floods, tropical storms, and so forth. About 3.6 billion people live in the area which are susceptible to climate change. And the data between 1998 and 2017 shows that approximately 160,000 people die per year due to extreme weather conditions. And this number is going to swell to about 250,000 by 2030. So the effects of climate change, as I said, is complex, and it could be because it affects in many different ways. You have the carbon emission is increased. You have increased level of particulate matter, greenhouse gases increase, and then you have it leads to depletion in the ozone layer in the summer months. Then you have wildfire. And all these things eventually lead to systemic inflammation, endothelial toxicity, and thrombosis. And then eventually culminating into cardiovascular disease and cardiovascular mortality. And just looking at the effects of thermal regulation on the cardiovascular disease, you can have these bursts of hot temperatures and cold temperatures, and both of them are capable of destabilizing the atherosclerotic plaque. The mechanisms are distinctly different. When we have the heat stress, it leads to peripheral vasodilation, but at the same time, we have fluid and electrolyte imbalance, and that increases the hypercoagulability and destabilizes the plaques. On the other side, when you have cold temperatures, extreme cold temperatures, that leads to vasoconstriction, it increases the blood pressure, and it also facilitates the deposition of cholesterol crystals in the atherosclerotic plaque. So that makes them more cholesterol-rich plaque and more prone to rupture. So when we look at the mechanism, it's not very different when we look at, especially in terms of heat stress. I mean, essentially, it promotes the prothrombotic events and pro-inflammatory events, which are the underlying cause of most cardiovascular disease. Then the climate change also leads to wildfire, and these wildfire incidence of these wildfire exposures has been increasing over a period of time as the global warming is affecting. And the effects of these wildfire are not restricted to the areas where the wildfire are, but it could be at very distal places. When we have the wildfire, you have the particulate matter, which is well known to cause cardiovascular and metabolic toxicity. But at the same time, you generate a lot of gaseous pollutants, especially volatile organic compounds, which can have deleterious health effects. And as said, the effects of wildfire could be at very distal places. And we had the first-hand experience of that last summer in Louisville itself, where the Canadian wildfire haze was affecting essentially the whole Kentucky area, especially Louisville. And this is the picture of from here in Louisville, downtown area, that haze covered the skies here in Louisville just because of wildfire in Canada. Another source of for generating these reactive chemicals and particulate matter is burn pit exposure, which is quite prevalent with the Army, and where in the combative areas, when they leave, they burn the whatever material they have, which they are leaving behind. And these burn pits have, because of the nature of this business, have various chemicals and woods and metals, and that generates a complex aerosol, which is very rich in the particulate matter and volatile organic compounds. And these exposures affect all the organs, including the cardiovascular system. In addition to these climate change associated effects, there are accidental exposures to hazardous chemicals, which could have long-term effects. Here in Louisville itself, from the local perspective, in 2012, we have a derailment of a railroad car carrying butadiene, and that really led to spillage of thousands of gallons of butadiene, and people were exposed to butadiene for almost 3 days, very high concentration. Then we saw that in East Palestine, Ohio last year, that train cars containing hazardous chemicals such as vinyl chloride were burned, and that led to this long-lasting exposure to particulate matter and organic chemicals. And very recently, this week itself, an apartment building caught fire in Louisville. And this is quite similar to exposure what we see in burn pit, because you have plastics, you have wood, you have all the organic material. So that generates the complex mixture. We know what the short-term effects of these complex mixtures are. And these include nausea, vomiting, and so forth. But we really don't know what are the effects of these acute exposure and then their chronic long-term effects. So that is a big question unknown. Then in addition to these, there are accidents which happened, which are perhaps at that time not known, where you are getting exposed to low levels of organic chemicals. And that happened in Camp-Lejeune, where more than 1.5 million military personnel were exposed for a long period of time to benzene and trichloroethylene. And they have a history of a variety of health effects, but unfortunately, it cannot be pinpointed what are the underlying cause and which chemicals are promoting it, whether it's a direct response or it's the autocrine paracrine effects, we don't know. But suffice to say that chemical exposure, both short-term and long-term, is deleterious for health overall, and especially for the cardiovascular system. So most of the data that is there in the literature in terms of environmental exposure and cardiovascular disease and metabolic disorders comes from the studies with particulate matter for the last 3 decades. PM exposure increases the prevalence of ischemic heart disease and stroke. And this is a slide from a review article published in Nature Medicine, where they showed that both, like doing the different modeling studies with integrated exposure response and global exposure response mortality. And in both the cases, they found there is a strong association of heart disease and stroke with the PM 2.5 exposure. And the studies from Sanjay Rajgopalan's lab in Cleveland show that PM has affects both the short-term effects as well as chronic exposure to PM is associated with a cardiovascular mortality. And they have segregated the data in 3 different zones, depending upon the nature of evidence. So there is a strong evidence here on the right side of the panel, as you see the blue triangle, in zone 1, which is very well characterized association of cardiovascular effects and all-cause mortality and cardiovascular mortality with the PM 2.5 exposure. And zone 2, then looking at what contributes to that is hypertension, diabetes, non-fatal MI, and non-fatal stroke and heart failure. And there are multiple studies have come out supporting these evidence, but this is still in process of getting quantified for various contributing factors. And then zone 3 is the thrombotic disorders and ventricle arterial and ventricle arrhythmias, where more data is needed to make a stronger case, but the evidence is there that there is some association of PM exposure with thrombosis. So when we look at the composition of PM, so it's composed of multiple things, and this is PM is just not one entity, it's a mixture of these particles coated with metals and volatile organic compounds. And as I have shown you in the previous slides, that the sources of volatile organic compounds are many, and that is the focus of our studies here at the University of Louisville. And if you look at the generation of volatile organic compounds from environmental exposure's point of view, that anytime you have combustion of a variety of materials, here we are looking at smoldering and flaming of plastics, plywoods, and cardboards, and so forth. You generate chemicals like benzene, acrolein, crotonaldehyde, and these volatile organic compounds are there in very large quantities, depending upon the chemicals which is being burned. Some of the other sources of generation of these volatile organic compounds is tobacco smoke, vaping, automobile exhaust, like because gasoline especially has benzene, ethyl benzene, toluene, xylene, and so forth. And the household products, several cleaning products, cosmetic products have a lot of these volatile organic compounds. These chemicals are also used in the industry, chemical industry, and in making the rubbers and plastic and so forth. And then heating of oil also generates a lot of these volatile organic compounds. So we are getting exposed to these chemicals not only through environmental pollution, but in the indoor air through cooking and household products, as well as through dietary sources. These volatile organic compounds are also very prevalent at Superfund sites, which are the hazardous waste sites. And the study done in the early 80s of 256 Superfund sites. They studied incidence of diabetes and stroke in people living closer to these 256 Superfund sites. Study of about 800,000 people showed that there was association of increased risk for diabetes and stroke in people living to these hazardous waste sites. And that was in part attributed to TCE, 1,3-trichloroethylene and benzene exposure. So building on these premise, in 2017, we established a Superfund Research Center here at the University of Louisville, which was funded by a National Institute of Health. And the objective of this center was to look primarily the effects of volatile organic compounds on cardiovascular and metabolic toxicity. It's a complex center in the sense that it has multiple components. We have biomedical projects, we have which are population-based studies and mechanistic lab-based studies. We have environmental science project, we have remediation strategies, and then we have multiple cores which complement the activities of the center. But in the interest of this group is the studies that we are doing with the volatile organic compound exposures with the human population. And this is the first prospective study looking at the effects of volatile organic compounds on cardiovascular disease. So in the first phase of our funding, we recruited 730 subjects to this study. And now we have expanded it to 1,200 participants to the study. And the goal is to look at a very comprehensive exposure assessment and look at the associations of various parameters of which are both functional as well as biomarkers of harm, which could be early detected and treated if possible to prevent the incidence of cardiovascular disease down the road. So for the exposure assessment, we have, and then we have potentially thinking in terms of some strategies for the intervention and quenching of these deleterious molecules to see if that affects the levels of overall exposures and improves the cardiovascular health. So for the exposure assessment, we have taken a multi-pronged approach. For the community level exposure, we have made a portable GCMS system which can be used for indoor and outdoor air measurements. We have also made specific VOC sensors which can capture these molecules in the indoor and outdoor air and can be brought back to the lab and analyzed. And then we are doing for large community level exposure assessment, we are looking in the wastewater, what are the levels of VOC metabolites, and what that way we can identify the potential sources at the neighborhood level, which are contributing to this environment exposure. For personal assessment, we established multiple platforms. Here you see this is one of the papers which came out last month, which was a cover story in the Environmental Science and Technology, where we developed a platform to look for the untargeted analysis of phase 2 metabolites of these environmental exposures. And then we have panel studies for targeted VOC metabolites that we measure in the urine. And from the health perspective, we are looking at various parameters, including the demographic information, physical exam, looking at the traditional risk factors for and housekeeping analytes, including the cholesterol, creatinine, CRP, ascorbic acid. And then we have a series of panels of biomarkers of harm that we measure in circulation. And then to account for tobacco exposure, because combustible cigarettes and even the vaping generates a large amount of these volatile organic compounds, we look at the cocaine level. To complement these human studies, we also have the animal studies, which test the plausibility that any given chemical identified in the human studies is sufficient to exert the cardiometabolic toxicity, and then to tease out the underlying molecular and cellular mechanisms. And some of the molecules that I'm going to share data with you today are depicted over here. Acrolein and crotonaldehyde are abundant in the combustible products. Benzene and xylenes are more petroleum products, but they are also generated during combustion. Vinyl chloride and trichloroethylene, these are organochlorine compounds used in the chemical industry, and 1,3-butadiene, which is used for making rubbers. So some of our data from the phase 1 of our study, and that again, although this is a prospective study, but data that I'm sharing today is from the cross-sectional study. In the first phase, the second visit is in process right now, so we will have data in time. But just a glimpse of some of the data coming out from this study shows that although there is a negative association of some of these VOC metabolites for benzene, crotonaldehyde, and acrolein with TNF-alpha, we have increased association with MCP1, which is a chemotaxic agent, IL-8, again associated with chemotaxis, and HSCRP, which is the marker of systemic inflammation. And then there's mild association with some of the white blood cells. And when we look at the functional level, what we find that metabolites of acrolein is associated with increased systolic blood pressure in our cohort. And these studies have subsequently been confirmed in the Jackson Heart study cohort, in which we measured VOC metabolites in 1,200 subjects, and I think in enhanced studies as well. And right now we are also doing studies with the MESA cohort, where we are analyzing 3,000 samples, complementing our human studies, using that as a confirmatory cohort, because they have 20 years data of atherosclerosis in that cohort. In addition to blood pressure, we also see that exposure to acrolein and 1,3-butadiene is associated with impaired vascular function depicted with RHI, which is essentially a reflection of arterial stiffness. Now, looking at, because we want to have an early detection of the exposure to these chemicals and their potential toxicity. So we are looking at the surrogate markers of endothelial toxicity. And in this case, we are measuring microparticles, which are shed from activated platelets or endothelial cells. And here we show the association with various volatile organic compound metabolites and the microparticles. The heat map on the left side shows the total sample size and then we can see similar response in the non-smokers, suggesting that it is not just coming from tobacco smoke, but environmental exposure is contributing to a great extent. And if we look at specifically at the endothelial microparticle and activated endothelial microparticles and dissociate the effect of age, then we find that for some of these molecules, age people have a stronger association than the younger people. When we look at the microparticles and their association with petroleum products, then we find that metabolites of benzene, toluene, ethylbenzene, and xylene. So most of them are essentially endothelial microparticles, and in some cases, platelet microparticles also associated with these kind of exposures, suggesting that exposure to gasoline product could be causing endothelial toxicity, which could have long-term effect on the cardiovascular health. And to confirm that the exposure to these chemicals is sufficient to cause the endothelial toxicity, we did the controlled exposure in experimental mice to these chemicals, in this case, benzene and xylene. And what we found that similar to human studies, exposure to benzene or xylene increased the levels of these endothelial microparticles and activated endothelial microparticles and platelet-derived microparticles in case of xylene. And our cellular studies show that exposure to metabolites of these molecules, especially for benzene, causes the apoptosis of endothelial cells. Then whenever we have endothelial toxicity to overcome the adverse effect, there is a release of angiogenic cells or endothelial progenitor cells from the bone marrow. So we looked at the potential repair capacity of the damaged endothelium by looking at these cells in circulation, and then this is the demographic information of the participants and which were enrolled for this study. And here the heat map shows that similar to microparticles, we see association of various classes of these angiogenic, circulating angiogenic cells in the total population as well as in the non-smokers. And especially if we look at some of the compounds of the VOCs that we think they are more associated with these kind of endothelial toxicity, we see that certain classes of these circulating angiogenic cells are associated with benzene, acrylonitrile, and ethylbenzene exposure. And controlled exposure, again, in mice shows that exposure to benzene is sufficient to deplete the levels of these circulating angiogenic cells, suggesting that it would compromise the repair capacity of the endothelium. So what are the long-term effects of these endothelial toxicity? I don't have time to go into details of all these studies, but our previous studies showed that these molecules, these volatile organic compounds, which are aldehydes like acrolein or N-crotonaldehyde, or which can be metabolized to aldehyde like benzene's metabolite mucoaldehyde, or which can have indirect effect in sense that that it can cause oxidative stress, and that generates reactive aldehydes. And so we use this approach to see whether aldehyde exposure is sufficient to exacerbate atherogenesis in mice, and whether the inhibition of aldehyde metabolism exacerbates the levels of these aldehydes and thereby exacerbate the toxicity, or if we can have chemical quenchers which can quench these aldehydes? Can you decrease atherogenesis? And those are the studies, outcomes of the studies that we did in the experimental animals. But what I'm going to share today is some of the newer data that we have generated using benzene as a prototype molecules. So exposure to benzene, these are animal studies. Exposure to benzene, we found that it causes insulin resistance, and that was oxidative stress dependent, as giving Tempol to decrease oxidative stress, decrease systemic inflammation, as well as oxidative stress and insulin resistance in mice. That's on the right panel. On the extreme left panel, you see that effect of benzene on atherogenesis in LDL receptor knockout mice. And in the aortic valve, there was increased accumulation of lipids in the lesions. And in the pressure overload induced cardiac dysfunction model, what we found that ejection fraction was significantly decreased in the mice exposed to benzene. So to look at to get some of the more mechanistic insight with the benzene exposure, we took the aorta with the atherosclerotic lesions and did the single cell RNA-seq experiment, try to identify what genes are associated and what are the cell types which are associated with the genes which are affected by benzene exposure. And what we found that there were 21 discrete population of cells that we could identify with these benzene exposure, which were in these aorta which were affected by benzene exposure. And here is the data that on the left side, we just see the Disney sort of analysis of the distribution of these cells. And you can see in the black circle that these are the T cells which are increased in number in benzene exposure. On the right side of the panel, it gives you the quantitative analysis of these benzene exposure. So what we found were three things which stood out. One, the T cell number was increased. Second thing was the number of proliferative cells in the aorta was increased. And the third thing was number of smooth muscle cells was increased. And when we look at the proliferative cells and stratify the data further, we find there is a massive increase in the T cells in the atherosclerotic plaque. And we are trying to tease out the mechanism right now. So we just got this week a grant funded from NIH looking at which is going to support these studies to have a mechanistic insight into the benzene-induced atherogenesis. And now then we wanted to see whether in terms of human exposures, just trying to reflect that, if you have established atherosclerotic plaque, even a short-term exposure, can that affect in such a way that it can destabilize the plaque? So here we did the CyTOF analysis of the aortic lesions of the benzene-exposed mice just after 2 weeks of benzene exposure. The previous was 24 weeks of exposure. And what we find here that similar to our RNA-seq data, even 2 weeks exposure to benzene was sufficient to increase the CD4 positive T cells in the plaque. So that makes this plaque more inflammatory and more cellular. And these are, I'm not going to go into details of all these RNA-seq data, but it's just showing you that the blue is the control mice, the red is the exposed mice. In several places, you can see an upregulation of the genes, and these are mostly genes associated with inflammation. Or in some cases, the genes are completely wiped out. So looking further closely into this process, we started looking at the cell chat analysis, looking at in the plaque itself, how these cells are communicating and what are the signaling coming in from a cell and signals going out from the cells, and how they could potentially be affecting these inflammatory processes in the plaque. And to cut the long story short, what we identify here, that IL-7 receptor is one of the targets, and then exposure to benzene completely wipes off the this IL-7 receptor in the atherosclerotic plaque. So these studies are ongoing, as we said, like we just got funded for that, and hopefully we will be able to come up with some more detailed mechanistic answers which can better explain the mechanisms of toxicity, which could be useful in coming up with some of the intervention strategies down the road. But going with the observation that we already have right now, I'm going to, in the next 5 minutes, I'm going to share some of these strategies which we are using at the Envirome Institute, not my lab per se, but our collaborators are using, building on the knowledge and database that we have generated over the years. And the first case here is using carnosine as a molecule for quenching the reactive molecules. So this is a dipeptide, beta-alanine and histidine. And what it does is binds to the across the double bond of these chemicals and tries to detoxify that. This is one of our studies in the animals where we showed that carnosine feeding decreases atherosclerotic plaque. It increases or it decreases the levels of protein acrolein adduct in the plaque. And if we overexpress carnosine synthase, which is a molecule which enhances the synthesis of carnosine in vivo, then and do a bone marrow transplant with the bone marrow overexpressing carnosine synthase, then we can decrease atherosclerotic plaque. So building on these, and then some of the mechanistic studies showed that overexpression of carnosine prevents a macrophage toxicity in vitro, and that is, again, we could recapitulate that in the atherosclerotic plaque and the right panel. As if you look in the upper one, in the wild type mice, you have a lot of apoptotic cells, and in the bottom one, you see like very minimal apoptotic cells are there. So building on these observations, there are 2 clinical trials which are ongoing in our institute. One is with the vascular function and thrombosis. This is led by Tim O'Toole, where he is looking at he has recruited 300 subjects, and we had the baseline measurements for the exposure assessment. And then they were giving carnosine for 3 months. And then after these 3 months of carnosine treatment, we brought those people back and again collected the data. And the primary target over here is looking at whether the carnosine feeding quenches these volatile organic compounds, and the readouts are platelet activation for thrombosis, systemic inflammation, and markers of endothelial toxicity. Then there is another study, and that is again an NIH-funded study, which is ongoing for 5 years now. And then the second study is looking at the effects of carnosine feeding on peripheral artery disease. And this study is led by Shahid Baba in our group. And essentially, same concept is whether quenching these reactivity of these molecules can have beneficial effects. And he got a very good score on his R01 grant this week. Hopefully he will be funded very soon. And the other study is what is very well known over here in the local population. Is called the Green Heart Project. Here, instead of doing the intervention at the individual level, we're looking at whether increasing vegetation can facilitate the removal of these environmental chemicals and whether that will have beneficial effects. And the premise of this study is built on a wealth of literature which is already there, which shows that proximity to green space is associated with this decrease in cardiovascular mortality. And in England, the rate of that cardiovascular mortality in least green areas was twice that in green areas. And then residential greenness is inversely correlated with ischemic heart disease and stroke. So in our study, this is, which is led by Dr. Arni Bhatnagar, this is the first prospective study in terms of a clinical trial sort of approach, looking at increasing vegetation has beneficial effects on cardiovascular health. And the idea is that here in the experimental design, the idea is that if you have increased greening, you can quench these environmental chemicals, and then that is going to be beneficial for the health. So in the South Louisville area, we have planted 8,000 trees in a neighborhood, and then a similar neighborhood without the plantation of trees serves as a control. And we have recruited subjects from these studies and doing a prospective study. And just a glimpse of some of the baseline study data. So we've seen that association between VOC metabolites and residential greenness over here. The some of these VOC metabolites, especially like benzene, the more you have greenness, the lesser the level of these aldehydes. So that means the system is efficient in terms of quenching these volatile organic compounds. And then looking at the sympathetic activation, because that could be one of the mechanisms by which it's affecting blood pressure and then vascular toxicity and so forth. So we find that, again, the levels of epinephrine metabolites in the urine is decreased if you have increased vegetation. And just, I don't have this, the crude data for this, but we just completed study looking at the effect, the levels of hsCRP as a marker of systemic inflammation, and see the association that if you have increased greenness, then you have the lower level of hsCRP, suggesting that you have lesser amount of systemic inflammation. Now, this has to be validated in the bigger sample size, and of course in the prospective studies. But it is very encouraging. And with that, I would want to wrap up and acknowledge the funding from the National Institute of Health. And this study was not possible without the active participation of members of the Superfund Research Center, Center for Cardiometabolic Science, and the Environment Institute as a whole. Thank you. And I will be happy to take any questions.
Dr. Srivastava, Dr. Hill, would you like to maybe go with a question for Dr.
Sure. How do I see who's going to?
Oh, you can, if you.
I do have a question.
Okay, go ahead. Go ahead.
So Sanjay, you talked about many interventions.
Just 3 chats. Well,
You talked about several interventions, one being harnessing, and then of course, going to a different level of intervention.
Yeah.
Planting trees. Harnessing's an amine-based scavenger, right?
right.
What's been done with like sulfhydryl-based scavengers? Of course, there's a lot of stuff on the market, methyl esters, ethyl esters that people buy and take.
Right.
Are they as potent?
So At the cellular level, we have seen, I mean, N-acetylcysteine is a very good quencher, right? I mean, glutathione conjugates these molecules, and N-acetylcysteine is like, but, and it decreases systemic oxidative stress at the animal level. But there hasn't been anything shown in the literature that I am aware of that these molecules have been tested for any beneficial effects. But one of the reasons is that they do multiple things. Like N-acetylcysteine is not only quenching these VOCs or like quenching these double bond, right, of these reactive carbonyls, but it does a lot of indirect effects, and that's where it becomes difficult in terms of mechanistic aspect what it is doing.
Right.
And Dr. Srivastava, we have a question from John Klein on
Yes.
the chat. He says, really enjoyed this talk very much. He wants to know, has this group examined any relationship between exposure response and immunogenetics profiles?
No, we have not looked at the genetic responses, and probably the reason is, for if you look at the genetic responses, you need much bigger population. So it would be difficult to do with the prospective studies and smaller sample size that we have. Once we have the data from the MESA cohort, which is 3,000 samples, and then the whole MESA cohort actually has 6,000 samples and a 20-year follow-up. That's one of the things that we want to do down the road, is immunophenotyping that cohort and looking at the association of various markers of exposures with the immune signals. But we are not there yet.
And he responded, said, yes, Mesa and Jackson cohorts may be sufficient.
Right.
Yeah. All right, I think that's all. So. Right, we can go ahead and
Thank you.
whoa, actually one more. Actually, this is from Dr. Cholera.
Mhm.
Got it just under the wire here. He says, Dr. Cholera says, great talk. Have you looked at interaction between PM 2.5 and VOCs synergistic? Is one worse than the other for MI? We have seen a relationship between PM 2.5 and high-risk coronary plaque on CCTA.
So in the population-based studies, it's very difficult to tease out the effect because PM itself has VOCs in there. PM is not a static entity. The composition of it is metals and VOCs and carbon and so forth. But we are planning to do some animal studies where we want to look at the effects of combined exposure to PM 2.5 and VOCs, and see if they have additive or synergistic effects, both in terms of vascular inflammation and plaque stability.
Say, no, Dr. Cholera, if you can interact if you'd like. I know I'll see him.
Yeah, no, I think this is a really great area of investigation, because we're seeing a higher number of myocardial infarctions, especially in younger people that live in areas where there's a high environmental exposure to both of these. And so I was wondering if, designing policy interventions is one more important than the other. And I know N95 masks, for example, will reduce the amount of PM 2.5, but the data is not very clear on, as you said, additive, synergistic, is one better than the other? Do masks protect one more than the other, et cetera? Certainly there's an association with blood pressure, and I think PM 2.5s in some studies have worse effect on increasing the systolic blood pressure than some of the other compounds. So it's just, whatever you think is perhaps more important, one versus the other, or we don't know. I think that's an area of investigation.
Yeah, but we don't know at this point, but if we have access to their urine sample, we can definitely look at the levels of some of these organic chemicals, their metabolites, and see if there's a similar correlation as you're seeing with PM 2.5. That will, again, strengthen the rationale to do the more targeted, perhaps, animal studies, which can show whether that exposure is sufficient to cause the cardiovascular disease, especially the plaque destabilization.
Fantastic. Yeah, again, very, very strong talk, great biological underpinnings. Congratulations, great work.
Thank you.
Yeah, one more question is from Dr. Don Castor in nephrology.
Uh-huh.
Says a great talk. Would love to look at environmental influence on autoimmune and chronic kidney disease in this population. Many of the inflammatory markers like MCP1 are implicated in autoimmunity.
I would love to collaborate with him. Yes, this is not, the kidney disease is not my forte, but definitely, I mean, I think similar biochemical mechanisms would be happening in other organs. We are working with Dr. McLean and Matt Cave looking at the liver, but we haven't looked in the kidney.
All right, I think that's it. Dr. Srivastava, she replied, said, you will be hearing from me.
Great.
Great deal. So, well, thank you again. This has been outstanding, and thank you for a great presentation. We got to over 100 participants on online, so we had a really good crowd, and really appreciate it. Thank you.
Thank you.
Thank you, everybody.